Hello everyone, my name is Dr. Srinath Satyanarayana and I work as Deputy Director at the International Union Against Tuberculosis and Lung Disease. At the outset, I thank National Institute for Research in Tuberculosis for inviting me to be a resource person for this course on tuberculosis. In this lecture, we will be discussing on the management of latent tuberculosis infection. Latent tuberculosis infection nowadays is referred to simply as TB infection. The concepts TB infection and latent tuberculosis infection are one and the same, and for simplicity I will continue to use the term latent tuberculosis infection throughout this lecture. This lecture on latent tuberculosis infection is divided into two sessions. In the first session, we will discuss about what is latent TB infection, how do we differentiate latent TB infection from TB disease, what is the estimated burden of latent TB infection globally and in India, what are the potential consequences of latent TB infection if not addressed, what are the options for addressing latent TB infection, who are the target population for latent TB infection detection and treatment, what are the steps involved in the detection of latent TB infection, what are the tests available for the detection of latent TB infection. In session 2, which I have not shown here, we will discuss about TB preventive treatment. Now, what is latent TB infection? To understand this, we need to know the spectrum of TB infection and disease. Once the Mycobacterium tuberculosis enters the human body, one of these 5 things can happen. The bacteria are eliminated either because of innate or acquired immunity, but in most cases it is contained in a localized area. In a small minority of the individuals, the infection may progress into subclinical or clinical disease immediately. The state in which the immune response of the body is successful in containing the infection in a localized area is called latent tuberculosis infection. Latent TB infection is defined as the state of persistent immune response to stimulation by Mycobacterium tuberculosis antigens with no evidence of clinically manifest active TB. On an average, it is said that about 5 to 10% of the people with latent TB infection develop TB disease during their lifetime, usually within the first 2 years after initial infection. Now, this slide explains the difference between latent TB infection, subclinical disease and active TB disease. There are 2 types of tests for latent TB infection. One is the tuberculin skin test and the other is the interferon-gamma release assay. We will discuss about these 2 tests in the later slides. For now, if these tests are conducted, persons with any of these 3 conditions will test positive, and therefore, these tests are not the right test to differentiate between these conditions. When we do sputum culture, people with latent TB infection will be negative, those with subclinical disease will be sporadically positive, and those with active disease will generally be positive. When sputum smear is conducted, it is usually negative, positive or negative in those individuals with active TB disease, but it is usually negative in those with latent TB infection. Regarding infectiousness, people with latent TB infection are never infectious. Subclinical are sporadically infectious, especially when they excrete the bacteria sporadically in their sputum. Those with active disease are considered to be infectious. Regarding symptoms, people with latent TB will never have symptoms. Persons with subclinical TB may have no symptoms or mild symptoms, and those with active disease may have mild or severe symptoms. Now, if you have to identify people with latent TB infection, these conditions must be met. That is, they must be positive on IGRA or TST, culture must be negative, sputum smear must be negative and should not be having any symptoms. Now, regarding the burden of latent tuberculosis infection at the global level, out of the 7 billion people, which is the estimated population of the world, about 2 billion people have latent TB infection. In India, it is estimated that about 30% of the population is infected with TB bacteria and may have latent TB infection. A small fraction but significant number of individuals, estimated to be about 6 to 7 million people, from this develop TB from this pool of 2 billion people. Therefore, latent TB infection is considered to represent the reservoir for tuberculosis from which TB cases keep trickling out. There are certain individuals who are at high risk of progression from latent TB infection to TB disease. These are the individuals with immunosuppressive conditions, HIV infection, radiographic evidence of prior healed TB, low body weight, that is 10% below the ideal, prolonged use of corticosteroids on immunosuppressive agents, silicosis, those who are recent TST converters. From a TB elimination perspective, since latent TB infection represents the reservoir of TB bacteria, preventing people from acquiring latent TB infection and preventing people with latent TB infection breaking down into TB disease are important. TB preventive treatment is one of the ways of reducing the breakdown from latent TB infection to TB disease, and therefore, treatment for latent TB infection is considered as one of the cornerstones of NTB strategy. Now, we will move to TB preventive treatment approach for preventing people with latent TB infection breaking down into TB disease. Here, we first identify the target population, rule out TB disease in them, conduct TB infection tests, identify those who are positive, provide TB preventive treatment to them and ensure TPT completion. The most important step in the whole cascade is to rule out TB disease. Now, once again, if you want, I can repeat this. Here we first identify the target population, rule out the TB disease in them. Ruling out TB disease is very important. Conduct TB infection tests, identify those who are test positive, provide TB preventive treatment to them and ensure TPT completion. And as I said, the most important step in the whole cascade for TPT provision is ruling out TB disease. Now, moving out to the target population for TB preventive therapy, as discussed earlier, nearly 2 billion people are having latent TB infection. It is technically and operationally challenging to identify and provide TPT to all of them. Currently, there are certain population subgroups who are at very high risk of development of TB disease and therefore, eligible for TB preventive treatment. These include people living with HIV, household contacts, other clinical high risk groups including individuals who are on immunosuppressive therapy, anti-tumor necrosis factor treatment, dialysis, preparing for organ or hematological transplantation and having silicosis. Now, regarding tests for detection of latent TB infection, there are no direct tests to detect the presence of TB LTBI in an individual. The screening tests rely on measurements of host immune response to bacteria. Accordingly, there are two tests: tuberculin skin test, also known as Monto test, Monto method, in which a purified protein derivative is injected into the host and then the individual's response to the solution of Mycobacterium tuberculosis complex antigens is measured. On the other hand, we have interferon-gamma release assays. There are two tests that are there. One is quantiferon TB gold in tube test and the other one is T-spot TB. Both these tests are based on assessing the production of interferon-gamma by the activated T lymphocytes in response to the TB specific antigen. It is of importance to note that neither TST or IGRA can separate latent TB infection from active TB disease. The availability and affordability of these tests will determine which will be chosen by the clinicians and program managers. Neither TST nor IGRA can be used to diagnose active TB disease nor for diagnostic workup of adults suspected of having tuberculosis infection. So, I repeat, both these tests cannot be used to diagnose active TB disease nor for diagnostic workup of adults suspected of having active TB disease. Now, moving on to the tuberculin skin test or Monto test, this measures cell mediated immune response. There are two strengths of purified protein derivative. One is the 5 tuberculosis unit PPD-S or the 2 tuberculosis unit PPD-RT23. These are injected intradermally into the forearm. Results, that is skin induration, is read between 48 to 72 hours after administration. A positive TST indicates TB infection. An induration of greater than 10 millimeter irrespective of the BCG immunization is considered as TST positive, and in certain individuals who are HIV infected or severely malnourished, an induration of greater than 5 millimeters is also considered to be positive. So, we must be careful in interpreting the tuberculin skin test result, as this test is prone for false positive and false negative test results. False negative test result can occur when an incorrect administration or interpretation of the test happens, improper storage of tuberculin in HIV infection, viral infections, measles, varicella infections, vaccinated with live viral vaccines, that is within 6 weeks people can tend to have negative TST. Malnutrition, bacterial infections such as typhoid, leprosy, pertussis can also cause negative TST, false negative TST. Immunosuppressive medications, neonatal patient, primary immunodeficiencies, diseases of lymphoid tissue such as Hodgkin's disease, lymphoma, leukemia, sarcoidosis, low protein states and severe TB. So, these are the conditions under which there can be a false negative TST result. And false positive TST results can also happen, and this can also result in wrongly interpreting somebody who is negative as positive TST. So, this can happen if there is incorrect interpretation of the test, if the reading is not taken within 48 to 72 hours, or if the measurement is incorrect, or infection with non-tuberculosis mycobacteria can also cause false positive TST. Now, regarding interferon gamma release assays, this is a in vitro blood test, that is blood test done in a laboratory. It measures in vitro release of interferon gamma by T cells in response to a well defined mix of synthetic peptides simulating antigenic proteins that are specific for tuberculosis. And there are two types of assays which are both FDA approved: quantiferon TB gold in tube test, which measures the interferon gamma concentration, and the other one is spot TB or T spot test, which measures the number of cells or spots producing interferon gamma. And then of late there is also another test which is there in the market, which is called the quantiferon gold plus. This is a new test which measures both CD4 plus and CD8 plus responses. There are certain advantages and disadvantages of IGRA. The advantages are it requires a single patient visit to conduct the test, the blood sample has to be collected. Results can be available within 24 hours, does not boost responses measured by the subsequent test, which means that when we are giving tuberculin skin test, there is always a possibility that the tuberculin can boost the immune responses once, and therefore, when we are repeating the tuberculin skin test again, the previous TST test can boost the immune response. So, that kind of boosting of immune response does not happen in IGRAs, and prior BCG vaccination does not cause a false positive IGRA test result, whereas in a TST that threat is always there, that a prior BCG vaccination can cause a false positive TST test. Now, the disadvantages of IGRA tests are the blood samples must be processed within 8 to 30 hours after collection, while white blood cells are still viable. Then errors can happen in the processing of white blood cells, and this can decrease the accuracy of IGRAs. The reagent costs of IGRAs are substantially higher, and then trained laboratory personnel and equipment is necessary for performing the test, and in general IGRAs are considered to be more costlier than tuberculin skin test. Now, test for latent TB infection, before we conclude, either a tuberculin skin test or an interferon gamma release assay can be used. Neither TST nor IGRA are to be used to diagnose active tuberculosis disease, nor for suspected workup of adults suspected of having active TB disease. Now, this concludes the first session. Thank you very much, and there are certain very important references that I have mentioned here, which you can read for further gaining information on tuberculin, whatever topic we have discussed so far. So, thank you.